Hi everybody. Today we're going to look at glycolysis overview and starting materials. Here are your learning objectives, so please take a moment and review these. The items here that are underlined and italicized are very important for you to memorize, and I can't test you on your memory of these, right? Because we're not together in the classroom, but I do promise you that if you take the time and you memorize the structures and you memorize the names of the enzymes, you will have an easier time moving forward in your career. So pause this, take a moment, and read through the learning objectives. Glucose is a very important molecule within the cell. It is a monosaccharide which can be used in anabolic processes that you see at the top of the image here. And in these anabolic processes, we can build large macromolecules which play a role that is structural or for long-term energy storage. Okay, so for example, glycogen in starch. Starch in plants, glycogen in people, and for us, glycogen in liver and skeletal muscle. Sucrose is another, made in plants molecule, but this is a disaccharide. Sucrose being glucose plus. As far as the structural carbohydrates that you see here, these are extracellular matrix or ECM, and we're looking here at the glycosaminoglycan component of the proteoglycans that are found in the extracellular matrix in us, and then we've got cell wall polysaccharides in plants, so things like cellulose. If you look at the bottom here, we're looking at essentially these are oxidative pathways which have to do with catabolism in general. And these are at the bottom. So the pentose phosphate pathway, also known as the pentose phosphate shunt, is able to oxidize glucose to make ribose 5-phosphate, which is, of course, necessary for nucleotide biosynthesis. And another product that we get out of this oxidative pentose phosphate pathway branch is NADPH, which is important for maintaining a reducing environment in the cell. Primarily, we need this NADPH to regenerate reduced glutathione, and we can also use NADPH for fatty acid synthesis. Okay, but our focus right now is going to be on glycolysis. And in glycolysis, we oxidize glucose to pyruvate, and we will also be forming some important compounds here like NADH and, of course, ATP. Where does glycolysis fit in the overall grand scheme of catabolism? And remember those buzzwords for catabolic pathways. They are oxidative. They rely on this cycling of the pool of NAD and NADH. So they generate NADH and they require NAD+, and we're able to recapture the energy from the bonds broken in catabolism to form high energy molecules like ATP. And of course, we are forming small molecules, and these tend to be convergent pathways. So we've covered all of the key buzzwords associated with catabolic pathways. Our focus is glycolysis, and glycolysis is here along this arrow. Glycolysis consists of 10 successive reactions, or would that be 11 successive reactions? There's a paper you're going to read in lab about that. So in glycolysis, we start with glucose, which is a 6-carbon monosaccharide, and we end up with 2 pyruvates, each having 3 carbons. Now, the pyruvate, the fate of the pyruvate varies by organism, cell type, and the availability of molecular oxygen. So, under aerobic conditions, which is our focus, that pyruvate will be transformed to acetyl-CoA, I should say decarboxylated, and then the remaining 2 carbons of pyruvate will be coupled to coenzyme A to form acetyl-CoA. And the enzyme that does that is pyruvate dehydrogenase complex. So I'm going to put PDH here, and we're going to focus on that. So again, just quickly right here. Pyruvate dehydrogenase complex transforms pyruvate through decarboxylation to acetyl-CoA, which has a high energy thioester bond. And acetyl-CoA flows into the citric acid cycle. So TCA, CAC, Krebs. When oxygen is not present, we got a choice here. So we have alcohol fermentation, and we've got lactic acid fermentation. Alcohol fermentation doesn't happen in us. Our cells don't run that. So we're not going to really worry about that now. That's what lab this week focuses on. But in us, lactic acid fermentation occurs. And in lactic acid fermentation, an enzyme called lactate dehydrogenase is going to convert pyruvate to lactate and in the process regenerate NAD+, so we could keep running glycolysis. And I'm going to go through, we're going to spend a lot of time looking at that. Okay, so going on. If we look at the differences here between respiration and fermentation, it's very clear. Respiration involves running of the electron transport chain, and in aerobic respiration, molecular oxygen serves as the terminal electron acceptor. In anaerobic respiration, we'll have an inorganic molecule that is not molecular oxygen. So NO3 minus, SO3 minus, etc. If we look at fermentation, fermentation means no ETC, right? No O2 required. So O2 might be present in the atmosphere and O2 might be available to the cells, but if you don't have the genes that code for the proteins that make up the enzymes in TCA or the complexes in ETC, you're not going to be able to do either process because you need the gene that codes for the enzyme that does the work. So then you would be running fermentation, right? And so at any rate, let's look at this. Glycolysis is important to both respiration and fermentation. The pathway of glycolysis is very highly conserved across all cellular life on the planet. Everybody needs to run some type of glycolysis. Now we're up to the reactions of glycolysis, and please do memorize the structures of all intermediates and the names of all of the enzymes, and of course, memorize the order of these reactions. So here you see the images from your book for glycolysis. On the left side, you can see the entire glycolytic pathway drawn out, showing you the names of the different enzymes, and you see very clearly the either Hayworth perspectives or just a standard representation of the structures of all of the metabolic intermediates. And you can see what steps require ATP, what steps are forming ATP, and where we have this step here is very important. Step 6, glyceraldehyde 3-phosphate dehydrogenase, is a step which requires NAD plus and generates NADH. This step also requires inorganic phosphate anion. This is a great part to look at to memorize the structures of these different molecules. And then we have the panel on the right. The panel on the right is showing you a different type of representation of molecular structure. And this is a fine structure to use. The panel on the right is useful because it talks about the chemical transformation which occurs between each of the intermediates. So this panel over here is pointing out to you the action of the enzyme on transformation of the intermediates. So what bond is broken? What bond is formed? What group has been transferred? And so this is more informative about the reaction mechanism. And if you want to learn the structures in this way, you can do that because it helps precise to you what has changed in these different structures, what has the enzyme done to process these different intermediates. But you should be able to use these structures as a baseline, right? If you memorize this, it's nice, it's clean, it's right angles, it's a linear, it's very simple, but you should be able to move from these representations of structure, which are kind of like a schematic diagram, back to a structure like this, which is more informative, right, than this. So for example, here you're seeing the cyclic pyranose form of glucose, and here you have a schematic representation. So glycolysis has 10 steps, and the first 5 steps are referred to as the preparatory phase. The last 5 steps are referred to as the payoff phase. So preparatory phase, ATP is used, and the last 5 steps, ATP is formed. Oh, and also NADH. And the NADH that's formed by glycolysis is able to support the electron transport chain. And of course, right, our electron transport chain makes a hydrogen ion gradient, which we then use to run ATP synthase and give us ATP. A little bit of a review here because you did this in your homework, so you should understand this concept, and I think that you do get this concept at this point. So oxidation, loss of electrons, loss of hydrogens. So oxidation means loss. And the carbons in glucose become oxidized through the process of glycolysis. Carbons of glucose most of them are alcohols, primary alcohols, and one of them is in the form of an aldehyde. And by the end of the process, all of the carbons of glucose will be fully oxidized and transformed to carbon dioxide. So there's 3 stages to glycolysis: collect the starting material, and then we have the first 5 reactions. Preparatory phase requires 2 ATP, and then we have the last 5 reactions which make 4 ATP. And so at the end of the process, we net 2 ATP for each molecule of glucose that enters into glycolysis. So step 1, we need to collect our glucose molecules and flow them into glycolysis. This graphic is showing you how glucose can be liberated from glycogen, which is a branched homopolymer of glucose. And we have alpha 1,4 linkages along the straight chains and alpha 1,6 linkages at the branch points. Glycogen phosphorylase is the enzyme which can remove glucose from glycogen, and it's not a hydrolysis using water, but it's a phosphorylysis using phosphate. And so it's inorganic phosphate that is used to break the alpha 1,4 linkage between these glucose residues. The product of this glycogen phosphorylase reaction is glucose 1-phosphate. And then we have these debranching enzymes. Now, the debranching enzyme is bifunctional. So it has 2 enzyme actions. It has a transferase activity and it has a glucosidase activity. The transferase activity is going to move the terminal 3 glucose residues to the non-reducing end of a straight chain of glucose. Do you see that? So these 3 residues here, we've broken. Transferase activity breaks alpha 1,4 linkage, and then we're going to transfer these 3 glucose residues to the end of a main chain. And then we have the glucosidase activity of the debranching enzyme, and the glucosidase activity breaks the alpha 1,6 linkage right here, and this is a hydrolysis reaction. So we've liberated then glucose in the process, which can go directly, of course, directly to glycolysis. And this glucose 1-phosphate molecules, they're going to go to glycolysis too, but we need to do some work on them first. So the glucose 1-phosphate molecules that came from glycogenolysis, see the PI? That's the inorganic phosphate that was used by glycogen phosphorylase. See glycogen phosphorylase here, endogenous glycogen. This is glycogenolysis. We end up with glucose 1-phosphate, and there's an enzyme called phosphoglucomutase. So phospho has a phosphorus. Glucose. Mutase, like mutate, change. So this enzyme is essentially going to move the phosphate from carbon 1 to carbon 6, but warning, it's not the same phosphate. And you'll see that when we look at the mechanism of these mutase enzymes. If we look at how the glucose derived from glycogen enters into glycolysis, it enters via this pathway and it goes into glycolysis as glucose 6-phosphate. Remember that. That's going to be important coming up. Now, there are other monosaccharides that can enter into glycolysis, but they need to be transformed first. For example, fructose. Fructose can be acted on by an enzyme called hexokinase, and this is going to generate fructose 6-phosphate, which enters into glycolysis. There is a second way that fructose can enter into glycolysis, and that is through the action of fructokinase. Kinases add phosphates, and fructokinase can form fructose 1 phosphate from fructose. And so we're going to be adding a phosphate here, right? That's fructose 1 phosphate. And then there's another enzyme called fructose 1 phosphate aldolase. Aldolases break the carbon-carbon bond between carbon 3 and carbon 4 of fructose. And so we end up with glyceraldehyde and dihydroxyacetone phosphate, also known as DHAP. And DHAP can enter directly into glycolysis. It's acted upon by triose phosphate isomerase, which is a glycolytic enzyme. And glyceraldehyde is going to be transformed to glyceraldehyde 3-phosphate by an enzyme called triose kinase. So let's address a couple other things here before we move on. Let's look at the approach to gathering starting glucoses from dietary glycogen and starch. So this is dietary glycogen, meaning that we've consumed it orally. This is endogenous glycogen, meaning that it came from liver or skeletal muscle. Dietary glycogen, along with starch, can be digested by alpha amylase, which is a hydrolase enzyme. And it uses water to break the alpha 1,4 linkages between the glucose residues in the glycogen and starch. And so we end up with glucose, which can, of course, right, goes into glycolysis because it's the first intermediate of the glycolytic pathway. If we look at sucrose, sucrose is acted on by sucrase, also known as invertase, and sucrase or invertase is able to break sucrose into glucose and fructose, and we covered how those monosaccharides enter into glycolysis. Do not worry about the mannose, that's fine. The glycosidic linkage in trehalose is hydrolyzed by trehalase, and we get 2 glucoses that go into glycolysis. Lactose. Lactose is broken down by an enzyme called lactase, also known as beta-galactosidase. And lactase breaks the beta 1,4 linkage in lactose to give us glucose and galactose. So the only other thing we need to look at here is how do we get the carbons of galactose to flow into glycolysis? And galactose needs to go through several transformations in order to be converted into glucose 1-phosphate. So how do we get glucose into the cell? Glucose is a large polar molecule. So we need a transport protein. So this is a glucose transporter to allow glucose to pass through the hydrophobic fatty acid tails of the membrane phospholipids. So here you see the GLUT2 transporter. This is a hepatocyte. So this is a liver cell that we're looking at. GLUT2 allows facilitated diffusion of glucose into and also out of the liver cell. And so it's not real sensitive to blood glucose concentrations. So glucose is able to move up or down its concentration gradient. So when glucose is high in the blood and low in the liver cell, glucose flows in. When glucose is low in the blood and high in the liver cell, glucose flows out through the same GLUT2 transporter. Now, the liver is special with regard to the first enzyme of glycolysis that's present. The liver has hexokinase 4, which is also known as glucokinase. More about that coming up. But this particular form of hexokinase is only in hepatocytes, only in the liver. So here you're looking at the first reaction of glycolysis in which glucose is transformed to glucose 6-phosphate. This requires ATP, and we will be releasing ADP, right? So this is a phosphorylation. If you look at the bottom panel, we have an enzyme here, which is the first enzyme of gluconeogenesis. And I know that this is a little bit out of order, but premonition here. Glucose 6-phosphatase is the undo enzyme of hexokinase 4. So this glucose 6-phosphatase enzyme removes the phosphate from glucose and allows glucose to pass out of the liver cell and into the blood.